Dominik Schwarz, University of Oxford, Distance Prediction for Flexibility Prediction. The motivation for our work lies in the fact that protein function is often coupled to switching between multiple structurally distinct states, and this is associated with flexibility of certain residues or certain regions. And knowledge about this flexibility or the protein structural ensemble would not only improve our mechanistic understanding of the protein, but could also enhance drug discovery studies, for example, in silico screening. And others have predicted ensembles of structures based on normal mode analysis, distance geometry, or coarse-grained MD simulations, but a dedicated sequence-based predictor is missing. The key research question of our work was whether rigid and flexible residue pairs have different co-evolutionary distance predictions. If we knew that, we could use these distance predictions to predict flexible residue pairs. And for that, we classified residue pairs into rigid and flexible by comparing two different PDB structures of the same protein and then analyzing the distance predictions of these different sets of residue pairs. Co-evolutionary information has been successfully used to improve protein structure prediction by improving the residue-residue contact prediction. This first has been done by avoiding transitive contacts, indirect contacts, through direct coupling analysis, and recently then by training neural networks on large sets of PDB structures alongside this co-evolutionary information. And then the most recent improvement has also been done by training neural networks, but instead of predicting the binary contact, yes or no, these networks were trained to predict the distance between two residues. And this yields a probability distribution over certain distance bins between the two residues. And we wanted to test whether these distance predictions can be used to predict flexibility of two residues, or then even to predict multiple relevant states of a protein. Every residue pair has a C-beta-C-beta distance. In the case of glycine, we took the C-alpha atom. And because we used two different PDB structures of the same protein, we also have the same residue pair twice, and we get two C-beta-C-beta distances. And from that, we can calculate the C-beta-C-beta distance difference. And the same is also true for physicochemical bonds between those two residues. And taking the criteria of the distance difference and the bonds, we classified residue pairs into rigid or flexible sets. And then we analyzed the probability distributions of these sets or of the residue pairs for the number of local maxima in the probability distribution. Local maxima in distance predictions we also call peaks, and for all distance predictions we analyzed this number of peaks, and some residue pairs have no peaks at all, some have one peak, and some have multiple peaks. And we then took the sets of residues and calculated the fraction that had 0 peaks, or 1 peak, or multiple peaks. And then we compared them between the sets of residues, and we found that rigid and flexible pairs have different fractions of these pairs. And first we compared the fraction of multiple peaks, which we assume is an indicator for flexible residue pairs. And we found that rigid residue pairs have a low fraction of this, with 22%. And flexible residue pairs have a higher fraction of those, with about 40%. And then we also looked at single peaks, which we think is an indicator for a rigid residue pair. And we also found that rigid residue pairs have a higher fraction than flexible residue pairs of these single peaks. And we also protein averaged this and calculated distributions, and we found that these distributions between rigid and flexible sets of residues are statistically significant. This is a short summary of our pipeline and of our main results. you can see some example distance predictions of 0, 1, or more peaks. And again, rigid and flexible sets of residue pairs were found to have different fractions of single, which are the blue ones in the stack bar plot, and multiple peaks, which are the red ones in the bar plot. So in conclusion, our hypothesis that flexible residue pairs have more local maxima or peaks in their distance predictions was supported, and also that rigid residue pairs have more single peaks in their distance predictions. And this result could potentially be used in multi-conformal modeling. Thank you very much for your interest, and also thank you to my sponsors. And if you have any further questions or comments, please come to the Q&A sessions. I will be there, or drop me an email.